Hi guys, how are you? Here is your own biology teacher Dr. Mahmood Hassan. Today we will study part 3 of amino acid and proteins. We will discuss the structure of proteins in detail. But before I continue, please subscribe to my channel Biology for All. The structure of proteins can be described at several levels of complexity. 4 levels of protein structure are commonly defined. These include primary structure, secondary structure, tertiary structure, and final structure of a protein called quaternary structure. In our previous lectures, we have already discussed that proteins are made up of amino acids. There are millions of types of proteins, but all these protein molecules are made up of only 20 types of amino acids. These 20 amino acids are alphabets of protein language, like 26 alphabets of English. The primary structure of proteins is determined by the sequence of nucleotides in the gene. Each set of 3 nucleotides, called a nucleotide triplet in the gene, codes for a specific amino acid. A polypeptide or protein molecule may contain several hundred amino acids linked into a long chain. A complete protein molecule may contain just one polypeptide chain, or it may have 2 or more chains which interact with each other. For example, the enzyme lysozyme has one chain of 129 amino acids. Insulin has 2 chains, one containing 30 amino acids and other containing 21. Now we look at the secondary structure of proteins. The amino acids in the polypeptide chain influence each other even if they are not directly next to each other. A polypeptide chain or a part of it often coils into a corkscrew-shaped structure called alpha helix. This secondary structure is due to hydrogen bonding between the oxygen of carbonyl group of one amino acid and the hydrogen of amino group of the amino acid 4 places ahead of it. Each amino acid has an amino and carbonyl group, and the figure shows that all these groups are involved in hydrogen bonding in the alpha helix, holding the structure firmly in shape. Hydrogen bonding is a result of the polar characteristics of the carbonyl and amino groups. Sometimes hydrogen bonding can result in a much looser, straighter shape than the alpha helix, which is called as beta pleated sheet or beta sheets. The overall 3-dimensional structure of a polypeptide is called tertiary structure. The tertiary structure is primarily due to interactions between the alkyl groups of amino acids that make up proteins mentioned as number 1 in this slide. Important to tertiary structure are hydrophobic interactions. Weak hydrophobic interactions occur between non-polar R groups. Although the interactions are weak, the groups tend to stay together because they are repelled by the water environment around them. Disulfide bonds, shown as number 2, are covalent linkages between sulfur-containing side chains of cysteines. They are much stronger than the other types of bonds. Third type of bonds are the hydrogen bonds that are formed between strongly polar groups, for example, amino, carbonyl, and hydroxyl group. In addition, ionic bonds form between ionized amine groups and ionized carboxyl group. They can be broken by the pH changes. Tertiary structure is the final structure of a protein that consists of a single polypeptide chain. However, when multiple polypeptide chain subunits come together, then the protein attains its quaternary structure. An example of quaternary structure is hemoglobin. The hemoglobin carries oxygen in the blood and is made up of 4 subunits, 2 alpha chains and 2 beta chains, each alpha chain containing 141 and each beta chain containing 146 amino acids. This is all for today, but the topic is still incomplete. So we will continue with the same topic in part 4, where we will study globular and fibrous proteins. We will also discuss the importance of amino acid sequence in a protein and also about the self-reactive proteins called prions. Please subscribe to my channel Biology for All and press the bell icon to get notified about the latest videos. Thank you.